Greetings, attendees to the International Material Research Congress. I am pleased to introduce Linda Gonzalez, a graduate of Biomedical Engineering Program at the Universidad Autónoma de Ciudad Juárez, who will present a very interesting topic entitled Bacterial Activity of Silver Nanoparticles in Drug-Resistant Bacteria, as part of the symposium Nanomaterials for Drug Delivery, Imaging, and Immunoengineering. Saludos a los asistentes al Congreso Internacional de Investigación en Materiales. Me complace presentarles a Linda González, egresada del programa de Ingeniería Biomédica de la Universidad Autónoma de Ciudad Juárez, quien presentará un tema interesante titulado Actividad bactericida de nanopartículas de plata en bacterias resistentes a fármacos, como parte del simposio Nanomateriales para la entrega de medicamentos, imágenes e inmunoingeniería.
I thank Dr. Cristian Chapa Gonzalez for the introduction. I will start with the presentation of the poster of my thesis, titled Bactericidal Activity of Silver Nanoparticles Systematic Review. First of all, bacteria are microscopic single-celled organisms that live in different environments, and they can be classified into gram-positive and gram-negative. The difference between these two is mainly in the composition of their cell wall. Gram-positive bacteria have cell walls made up primarily of a substance known as a peptidoglycan, and these bacteria stain purple after instead gram-negative bacteria that have cell walls with only a thin peptidoglycan layer, and also have an outer membrane. And gram-negative bacteria stain red or pink after gram stain. So the resistance occurs when bacteria change or mutate, so antibiotics no longer work to treat infections. When the strongest survive, so the bacteria already have resistance to antibiotics in their genetic information, and they will pass that gene to the next generations of bacteria. Therefore, unconventional methods are being sought that can confront bacterial resistance, such as silver nanoparticles. The mechanism of action is shown in this image, where the silver nanoparticle first binds to and providing cellular respiration. In the same way, the nanoparticle prevents the cell from taking nutrients, and finally, this penetrates the cell membrane, breaking the DNA chain and preventing its replication. Picture of the issues at stake, the question is formulated: in what type of drug-resistant bacteria, gram-positive or gram-negative, are silver nanoparticles more? The main objective of this project is to determine the type of drug-resistant gram bacteria in which silver nanoparticles are most effective as bactericide. The methodology used was, first of all, the select database search. I used ScienceDirect, PubMed, and EBSCOhost. Second of all, declaration of search terms with booleans. Then compilation of articles according to the data of interest. Then the data tabulation process, and evaluation and analysis of collected data. And finally, conclusions. Through the PRISMA diagram, the number of records in the searches were identified. From the ScienceDirect database was 1,000 281 articles. From PubMed was 50 from EBSCOhost. Of all these records, only 6 were included in this in the study that meet all the requirements. And the main characteristics analyzed in the sources of evidence were, first of all, synthesis, type of a study, nanoparticle size, incubation time, type of test, and finally, test results. The common objectives of the 6 selected articles were to successfully synthesize silver nanoparticles and use them. Finally, all the studies agree that silver nanoparticles are effective against both gram-negative and gram-positive bacteria. However, gram-negative bacteria were more sensitive to silver nanoparticles, and this may be due to the difference in the cell wall, on the cell wall of each strain. And the conclusion of this systematic review are summarized in silver nanoparticles is a method with a great future, sustainable and more economical. Also, records are needed in studies that suggest or mention a possible generation of bacterial resistance to silver nanoparticles. And finally, the study also indicates that these metallic nanoparticles have a greater effect on gram-negative bacteria due to the composition. Thanks a lot for your attention.